Cloning was invented during the 20th century. The first ever demonstration of artificial embryo twinning was in 1885 by Hans Reich in Germany, who reproduced a sea urchin. The following years, many biologists continued cloning all around the globe. Cloning is the reproduction of organisms without sex. It is the joining of genetic material from an adult organism into an embryo that may rise into a new adult organism. Offspring are genetically identical to their single parent. Researchers have cloned a wide range of biological materials, including genes, cells, tissues, and even entire organisms. In 1996, scientists succeeded in cloning the first mammal from a cell taken from an adult animal. After 276 attempts, Scottish researchers finally produced the famous Dolly, the lamb from a somatic cell in a 6-year-old sheep. Until today, there is no solid scientific evidence that anyone has cloned humans yet. Cloning humans and other primates is more difficult than in other mammals. How does it work? Cloning can happen naturally and inside the lab. There are two ways to make a clone in the lab: with artificial embryo twinning and with somatic cell nuclear transfer. Twinning relies on the merging of egg and sperm. It happens in the first days while the embryo is made of just a small number of unspecialized cells. Each half of the embryo continues dividing on its own, ultimately developing into separate, complete individuals. Since they develop from the same fertilized egg, the resulting individuals are genetically identical. Artificial embryo is a technique that mimics the natural process of creating identical twins. It is carried out in a petri dish, instead of inside the mother. A very early embryo is separated into individual cells, which are allowed to divide and develop for a short time in the petri dish. The embryos are then placed into a surrogate mother, where they finish developing. Again, since all the embryos came from the same fertilized egg, they are all genetically identical. The somatic cell nuclear transfer consists on transferring a somatic cell, which is anything that is not sperm or egg, into an egg cell. It starts with the isolation of a nucleus from an egg cell. Then, the nucleus of the somatic body cell with desired genes is removed and transferred to the first enucleated egg cell. After a couple of chemical tweaks, the egg cell with its new nucleus starts behaving just like a freshly fertilized egg. It develops into an embryo full of stem cells, which is implanted into a surrogate mother. Finally, an exact genetic copy or clone of the mother is created. This was the method used to create Dolly the sheep. Cloning is important for many reasons, including advances in medicine, the faster production of livestock, the improvement of crops, and for the use by the police. Another way we can use this technology is if we can use cloning for creating new human tissues. This would be therapeutic cloning, which has a great use in healing diseases such as diabetes, Alzheimer, and Parkinson.